Following the consumption of a meal, the body breaks down and absorbs the dietary lipid and occurs in the small intestine, but also involves other aspects of the GI tract, including the stomach, liver, pancreas, and to some extent, colon. To put it on a scenario, the small intestine is considered the factory. So this is the scenario. So we have raw materials that are outside of our body. So these raw materials are the food that we intake. To put it on a scenario, the small intestine is considered as a factory which is producing particular products which need to be distributed from the factory to points of sale. These points of sale are the cells throughout our body. These products are broken down, repackaged, as they are absorbed into small enterocytes and then repackaged into triglycerides and cholesterol esters. These dietary lipids are then repackaged into chylomicron. These dietary lipids are then packaged into chylomicron, which are produced in the integral sites. The lipids are now sent out through the circulatory system via the lymphatic system to various tissues such as muscle and adipose tissues. Once the chylomicron has left remnants, they are recognized by receptors at the liver and removed from circulation. So to specify the exogenous pathway, it is a long-chain fatty acid and are esterified into triglycerols in the gut and packaged into chylomicrons, which contain ApoB48 and are synthesized and secreted into blood via the lymphatic circulation. ApoC, ApoE, and cholesterol ester are acquired from HDL in circulation. ApoC activates lipoprotein lipase, which catalyzes the hydrolysis of triglycerols, and ApoC is transferred back to HDL. The chylomicron remnant is taken up by the ApoB48 remnant receptor in the liver. Hepatically derived lipid, which is being packaged into the VLDL particles and then transported throughout the body to deliver both triglycerides and to some extent cholesterol to various cells and tissues. A scenario where a factory is capable of taking raw material from circulation, which include triglyceride and cholesterol. Repackaging this into the transport lipoprotein or the very low density lipoprotein packaging. This into VLDL particles and shipping it out in circulation to distribute it slip through the various cells. Podocytes are capable previously produced triglyceride and cholesterol or producing its own fatty acids, triglycerides and even cholesterols, and then packaging them into the LDL particles for transport to other cells and tissues. Lipoprotein particles are delivering the lipid to the various cells. Once the triglyceride and some of the cholesterol has been adequately distributed to these tissues, these VLDL particles have now gone through a transition to LDL particles, which are very poor in triglyceride content but high in cholesterol. Ideally, these LDL particles are then removed from circulation by the liver and the process can start over again. With these LDL particles being broken down their constituent parts and repackaging so that this particular cycle occurs again. To specify endogenous pathway, triacylglycerol and cholesterol synthesized in the liver are secreted in VLDL, which contains ApoB100. ApoC and ApoE and cholesterol ester are required from HDL in circulation. APOC activates lipoprotein lipase, which catalyzes the hydrolysis of triacylglycerol. APOC is transferred back to HDL. The remnant particle, or IDL, if it contains APOE, can be taken up by the APOE or remnant receptor, or IDL without APOE becomes a substrate for hepatic lipase and is converted to LDL. LDL is removed by ApoB100 receptors, which are mainly expressed in the liver. Cardiovascular diseases is one of the leading causes of mortality worldwide, and its chief underlying cause is atherosclerosis, which is a chronic inflammation of the arterial wall. The wall of an artery consists of three layers: the adventitia, the tunica, and the intima, where stages of atherosclerosis begin. At normal concentrations in the blood, low-density lipoprotein molecules can pass in and out of the intima. However, when in excess, these molecules can accumulate within the subendothelial cell layer, where they become highly susceptible to oxidation. This results in the formation of oxidized low-density lipoprotein molecules, which are toxic and consequently trigger an inflammatory response. Endothelial cells in the intima respond by increasing their expression of various leukocyte adhesion molecules, particularly VCAM1 on its cell surface. Local tissue also secretes several chemokines that directs monocytes to the site of inflammation. Monocytes adhere to VCAM1 and migrate between the intact endothelial cells in order to penetrate into the tunica intima. Once resident in the intima, the monocytes acquire the characteristics of the tissue macrophage, which then engulf the trapped oxidized low-density lipoprotein molecules. This process gives to an arterial foam cell in which the macrophage becomes so packed with fatty droplets. Circulating cytokines and growth factors may also promote the migration of vascular smooth muscle cells through the internal elastic lamina, where it forms a tough fibrous cup surrounding the atherosclerotic plaque. This plaque results in the narrowing of the vessel and can potentially rupture, causing the release of tissue factors that may result in the formation of a blood clot.